Although all bacteria have a cell wall containing peptidoglycan, there are two distinct structural types of cell wall known as gram-positive and gram-negative. The names derive from the Danish scientist Christian Gram, who in the year 1880 derived the rapid staining technique which could differentiate bacteria belonging to one of the two basic types, and this technique was known as Gram staining. For any stain, you must first smear the substance to be stained into the slide, then heat to fix it the bacteria on the slide. There are 4 steps, 40 grams to stain. The first step is to pour the crystal violet stain and wait for 60 seconds. Then you wash off with water and flood with iodine solution for another minute. Wash off with water, then decolorize with 95% alcohol. Then finally counterstain with safranin, which is a red dye. You wait for 30 seconds and wash off with water. When the slide is studied microscopically, cells which absorb crystal violet and hold it will appear blue. These are called gram-positive organisms. However, if the crystal violet color is washed off by alcohol, these cells will absorb the safranin and appear red. These are called gram-negative organisms. The different stains are the result of differences in cell walls of gram-negative and gram-positive bacteria. Gram-positive cell walls are relatively simple structure comprising of several layers of peptidoglycan which is connected to each other by cross linkages to form a strong, rigid scaffolding. In addition, they contain acidic polysaccharides called teichoic acids, and these contain phosphate groups which impact an overall negative charge on the cell surface. Gram-negative cells have a much thinner layer of peptidoglycan. This makes the walls less sturdy. However, the structure is made more complex by presence of a layer of lipoprotein, polysaccharide, and phospholipid known as the outer membrane. Instead of 2 layers of phospholipid, it has only 1, and the outer layer being made of a lipopolysaccharide. This outer layer has 3 parts: the lipid A, copolysaccharide, and an O-specific side chain. The lipid A component may act as endotoxin which, if released to bloodstream, can lead to serious conditions such as fever and toxic shock. The O-specific antigens are carbohydrate chains whose composition varies between strains of the same species. Proteins called porins incorporated into the outer membrane and penetrating its entire thickness form channels, and these channels allow the passage of water and small molecules to enter the cell. The outer membrane plays no part in cell respiration, and some bacteria, mostly of the gram-negative type, have a periplasmic space between the plasma membrane and the cell wall. And this space is the site of metabolic activity and contains a number of enzymes and transport proteins. Gram-positive cells have 2 layers, the inner cytoplasmic membrane and the outer thick peptidoglycan, while the gram-negative cells have 3 layers, which is the inner cytoplasmic membrane, thin peptidoglycan layer, and an outer membrane with lipopolysaccharide. Gram-positives have a low lipid content, while the gram-negative have a high lipid content. And gram-positives have no endotoxins, while the gram-negatives have lipid A, which is an endotoxin. Gram-positives have no porins, while gram-negatives have a porin channel. And gram-positives are vulnerable to lysozyme and penicillin attack, while gram-negatives are resistant to lysozymes and penicillin attacks. The gram-negative cell envelope has S layers, not including the periplasmic space. The thin peptidoglycan layer does not contain teichoic acid, although it does have a small helicoprotein known as murine lipoprotein. This lipoprotein is important because it originates from the peptidoglycan layer and extends outward to bind the unique third outer membrane. This last membrane is similar to the cell membranes in the cells, that is composed of 2 layers of phospholipid bilayer with hydrophobic tails in the center. What makes it unique is that the outermost portion of the bilayer contains lipopolysaccharide or LPEs. The differences between gram-positive and gram-negative organisms result in a valid interactions with the environment. The gram-positive thickly meshed peptidoglycan layer does not block division of low molecular weight compounds, so substances which damage cytoplasmic membranes such as antibiotics and detergents can pass through. However, the gram-negative outer lipopolysaccharide containing cell membrane blocks the passage of these substances to the peptidoglycan layer and a sensitive inner cytoplasmic membrane. Therefore, antibiotics and chemicals that attempt to attack the peptidoglycan cell walls such as penicillin and lysozymes are unable to pass through. Interestingly, the crystal violet stain used for gram staining is a large dicomplex and is trapped in the thick cross-linked gram-positive cell wall, resulting in a gram-positive blue stain. And the outer lipid-containing cell membrane of the gram-negative organisms is partially dissolved in alcohol, therefore washing out the crystal violet and allowing saffron counterstain to take.